Hello, my name is Joanne, and we're going to be talking about G protein coupled receptors. There are three main categories of cell surface receptors: G protein coupled receptors, ion channeled coupled receptors, and enzyme coupled receptors. G protein coupled receptors are the most widely used transmembrane receptors. These receptors are responsible for carrying the signal into the cell by a series of modified proteins. When a signal molecule is bound to it, the receptor changes the target molecules directly. G protein coupled receptors are sensitive to a wide range of cellular signals, such as hormones, neurotransmitters, and paracrine molecules. GPCRs are key components to every aspect of the cell communication. They are also important in the signal molecules that interact with and bind to GCPR. These are transmembrane proteins that are found at the cell surface that were synthesized at the ER. The synthesis includes the threading through the endoplasmic reticulum via start and stop transfer sequences, as well as the shipping and processing at Golgi. When a signal molecule binds to its receptor, the receptor will change its shape. The change of shape allows for the activation of G proteins that are associated with the receptor at the face of the membrane. All types of G proteins bind to GTP and GDP. Every G protein has an alpha, beta, and gamma subunit. The alpha subunit will bind to the GDP, which will allow for the G protein to be dormant. When the receptor has a secondary change in shape, the G protein decreases its affinity for GDP and increases affinity for GTP. Therefore, the G protein will let go of GDP and will bind to the GTP, making the protein active. Once activated, the alpha subunit can alter target molecules, and the remaining beta-gamma complex will do the same, independently initiating the signal cascade. The system is now on, but signals must be turned off. This is not an issue for G proteins because it has a GTPase activity that allows it to hydrolyze GTP and GDP, resulting in an inactive form. The subunits will reassociate and translocate back to the receptor. This process will repeat when the new cell receives a signal. G proteins can activate membrane-bound enzymes or channels, open ion channels, and have an effect on GPCR-controlled enzymes. The G protein affect enzymes at the cell membrane, and these tend to have more complex and cascading effects. The two most common enzymes being activated by the G proteins are adenylate cyclase and phospholipase C, which are activated by different GPCRs. Adenylate cyclase catalyzes the biochemical reaction to convert ATP to CAMP. CAMP carries the message into the cell that activates kinase A. This protein phosphorylates other target molecules to result in inhibition or activation of a given physiological response. A second messenger in the common GPR systems is calcium. Calcium uses a membrane component known as PIP2. Phospholipase C splits PIP2 into two other molecules, IP3 and DAG. IP3 leaves the membrane and travels to the cell's interior. DAG membranes will activate protein kinase C phosphorylation for other target proteins, including the calcium channels that allow calcium into the cell. IP3 goes to the endoplasmic reticulum and triggers a release of calcium. That calcium then binds to PKC, phosphokinase C. This is making it more active. Calcium also binds to calmodulin, making it active. When calmodulin is active, it activates another kinase, which phosphorylate target proteins that go on to do other things in the cell as well.